As aging progresses, the body's intricate network for maintaining energy balance becomes increasingly dysregulated. Among the most critical and overlooked components of this system is the hypothalamus, a small but powerful brain region that integrates signals related to hunger, satiety, thermogenesis, and nutrient availability. The hypothalamus plays a central role in appetite regulation, and its dysfunction contributes significantly to age-related weight gain and metabolic imbalance. Emerging evidence suggests that autophagy within the hypothalamus is a key determinant of its regulatory capacity, and this process, like many others, declines with age. Nicotinamide mononucleotide (NMN), a precursor to NAD+, has drawn growing attention for its potential to restore cellular housekeeping functions, including autophagy, in metabolically active tissues. Investigating whether NMN can reactivate hypothalamic autophagy and thereby restore appetite control represents an exciting frontier in metabolic aging research. The hypothalamus responds to a variety of internal cues, including circulating hormones such as leptin, insulin, ghrelin, and nutrient-derived molecules like glucose and fatty acids. Specialized neurons, particularly those in the arcuate nucleus, AGRP and PUMC neurons, interpret these signals and adjust feeding behavior and energy expenditure accordingly. As organisms age, this sensory system becomes blunted. Leptin resistance develops, satiety signaling weakens, and orexigenic, appetite-stimulating pathways become overactive. A growing body of research now shows that one of the root causes of this dysfunction is impaired hypothalamic autophagy. In healthy young brains, autophagy helps regulate the number and sensitivity of key metabolic receptors, removes damaged mitochondria and misfolded proteins from neurons, and maintains synaptic plasticity needed for adaptive energy sensing. With aging, autophagic flux in the hypothalamus declines, leading to cellular stress, inflammation, and disrupted neuropeptide signaling. This is where NMN enters the equation. By replenishing NAD+ levels, NMN activates sirtuin pathways, particularly SIRT1, AMPK, and other nutrient-sensing mechanisms that are known to regulate autophagy. In the hypothalamus, SIRT1 acts as a metabolic switch that enhances neuronal resilience and supports the autophagic machinery. Experimental models suggest that boosting NAD+ through NMN can reduce hypothalamic inflammation, improve mitochondrial function, and re-engage autophagy in energy-sensing neurons. One of the key downstream effects of this restoration is the rebalancing of AGRP and PUMC neuron activity, the primary drivers of hunger and satiety, respectively. By fine-tuning their responsiveness to circulating signals, NMN may help suppress excessive appetite and restore the brain's capacity to maintain a healthy body weight set point. In aged or diet-induced obese animal models, NMN treatment has shown promising outcomes related to hypothalamic function. Studies indicate reduced microglial activation, a marker of neuroinflammation, improved leptin sensitivity, and upregulation of autophagy-related genes in hypothalamic tissue. These changes correspond with normalized feeding behavior, reduced fat mass, and improved glucose tolerance. Such results support the idea that the hypothalamus is not just passively affected by aging, it is a modifiable target whose function can be partially restored through metabolic interventions like NMN. In essence, NMN helps reestablish a clearer dialogue between the body and the brain, one that is necessary for the nuanced regulation of appetite and energy balance. Another important dimension is the prevention of neurodegenerative changes that can occur in hypothalamic circuits with age. Autophagy plays a crucial role in neuronal health by removing dysfunctional organelles and protein aggregates, which, if left unchecked, can lead to cell death and impaired signaling. NMN's ability to boost autophagy in hypothalamic neurons may also serve a neuroprotective function, preserving the integrity of appetite-regulating pathways before irreversible damage sets in. This effect could be especially valuable in older adults, where unintentional weight gain due to impaired central control often leads to insulin resistance, cardiovascular risk, and reduced mobility. While direct evidence from human trials remains limited, the mechanistic clarity observed in preclinical studies provides a compelling foundation for further exploration. If NMN can indeed restore hypothalamic autophagy and recalibrate appetite control in older individuals, it could offer a novel strategy to address one of the most stubborn aspects of metabolic aging, the tendency to gain weight even without a dramatic increase in food intake. Unlike stimulant-based appetite suppressants or blunt hormonal interventions, NMN operates by rejuvenating the core cellular processes that underlie the brain's energy regulation system. Taken together, the hypothesis that NMN can bridge the gap between aging, hypothalamic dysfunction, and disordered appetite finds strong theoretical and experimental support. By enhancing autophagy within hypothalamic neurons, NMN not only improves metabolic signaling and neuronal viability, but may also help re-anchor body weight regulation to a healthier baseline. This tissue-specific approach reinforces the idea that aging-related obesity is not just a peripheral problem of fat accumulation, but also a central issue of impaired energy sensing, and that the brain itself, through mechanisms like autophagy, can be retuned to better serve the body as it ages.